السلام عليكم ورحمة الله وبركاته. علينا وعليكم. الحمد لله بعلمين. How are you?
Alhamdulillah. If we have to make a wuzu here, what's the best way?
There's a washroom right beside on the right side. Washroom in this way,
On the floor?
on this floor, down that side. And you'll see the right side for the brothers.
How you can wash your feet?
They have wudu station as
Oh,
well.
they put a wuzu station. Nice. And if we have to check the timing of the salah, can we do the online check or something?
Yes, they have a digital. They have online if you check on phone or even on
What do you have to
timetable.
type?
You
www.
you could do islamicfinder.ca.
No, especially for this here.
There they have a clock.
I know it's a digital clock, but it's already hooked up with the internet or something more, right?
They do it as calculations, or let me see. I know it's a specific timing.
Like other salah, where Abu Bakr, Huzaifa, all that have their website and times are displayed
That's
nicely. Because time is changing. Right?
Yes.
It's easy to catch up.
Yes, yes.
So if I'm here, I have to come and check and then go back.
I see. Okay.
Or if I'm nearby and I know my salah is missing, so I can pick it up here.
That's true. Well, or inshallah to come by here.
Perform here.
If you search,
are you looking any device? Let me know. We can pay. Just install it. Like small device and timing, and also have a provision to connect with the internet.
I believe it does have them. It already has the time.
Internet connection.
Internet. Let's see.
That's what I'm asking. So at least any time you can on board. You can quickly check. You're nearby. Boom. You can come. And we're gonna be helpful further. Let me know the price. Or normally university pay for these kind of stuff. I don't know.
I don't know if the other one is different because they have the times as well, and it changes every minute or
yeah, but it's only local, right? You can only see.
Yeah.
When you're here, then you can see. But it's not on the smartphone.
I see.
How many halls like that? Yes. My son told me something there too. On the other side. It's only one here.
Maybe he has found a spot to create. I don't know.
Check it again. So you are saying only one?
There is only one here. There is technically one down there in another building called environmental science.
Yeah, I know.
But it's small. Yeah. And then there's one in the Sam Ibrahim building on the 5th floor.
The new building.
The new building.
And nothing on this backside.
Oh, not to my knowledge. Maybe it could be like a small area that he's spraying. Hello, everyone. How's everything going?
Good, how are you?
Sorry, I missed my my computer was on mute, so I couldn't actually hear you.
Oh, I said good, how are you?
I'm doing great as well. I'm in the midst of opening up the slides for us to go over the review. So maybe just to briefly ask, has everyone had a chance to or had the opportunity? Has everyone had the opportunity to have a look at the slides, or sorry, the review from the past? It was posted on YouTube as well. And apologies if also you can't hear if there's a bit of background. I'm in the student center. I'm just making sure I have everything that I need to show you guys. Give me another minute. But everything else okay? Understanding wise the material for the lab exam? Yeah. And if you need to, you can also use the chat, but I At the moment, as I open slides, I can't really tell. Give me just a moment, folks. If you have friends, people from BIOA02 that wanna, inshallah, get a heads up. And I realized how the zoo was a separate venture, right? Because we didn't have a PowerPoint, which is making sense to me now. So let's go over the material. And let's go for it. Okay. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. So you're gonna go to your lab manual if you want to for reference, page 55. So nice, we're having some folks with different symbols as names. We have a period. We have what looks like a koala bear. But you guys can mention your names if you wish to. And so that way I could get a good idea as to who we're with today. Wanna share your names? The one with the period, the one that has a cow or a koala bear. A koala as a symbol. Oh, Dorlin, okay. And the one with the period. Oh, I'm noticing chat is open. Let me see. Okay, that's okay. No problem. No problem, folks. If I can't also get access to the chat as I talk, feel free to let me know if anyone sees, inshallah, like any points that maybe I'm missing out. But I'm gonna go over a bit of the practical. So we went from week 4 and then on to on Zoom, right? So أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. So if we have a look and recall back into the past, and by the way, you probably may have noticed that some of my recordings are made so that you guys can look also into the future for your lab exam. So if you want to have like a look at the same channel, it's called Our Golden Age, where I post my content from the practicals, so like recordings if you wish. But essentially, when it comes to lab 4, we dealt with thermal regulation. Do we recall the definitions? There were 4 different types: endotherm, ectotherm, homeotherm, and poikilotherm. Anyone remember the differences between some of them? And feel free to share it out loud or text as well. Yes, so to repeat the question, there are 4 different definitions I mentioned. No problem. 4 different definitions, rather, um, of the different thermoregulatory paths or characteristics/traits that organisms take. We had endotherms, ectotherms, homeotherms, and poikilotherms. And so as we see from Joshua, endotherms self-regulate and ectotherms environmentally regulate. That's sound. That's a good, like brief, succinct definition. In terms of endotherms, and so referring to the difference or the categorization of endotherms versus ectotherms, they focus on the source or the origin of thermoregulation. So endotherms have it through metabolic pathways that are internal, right? So think about chemical reactions, metabolism, as we saw, or the heart rate with Daphnia, poikilotherm, which is a different topic altogether. But essentially, you have endotherms like ourselves. Then you have ectotherms, right? So think about sometimes this is the case of cold-blooded animals or mammals, but let's say of trying to think. A good example is the Daphnia, actually, right? They environmentally regulate their temperature based on the temperature of their environment. So the temperature of, let's say, water, where the Daphnia water fleas reside, is where they essentially may essentially have changes or may respond accordingly in their in changes or fluxes in their core temperature. Um, let's see, endo is pretty much heating their own body. Ectotherm. So point in mind, exo is different from ecto. Ecto is for biology and exo is for chemistry. So sometimes I know people may have gotten this mistaken, but bear that in mind because if there's, let's say, like fill in the blank, you want to make sure you're saying the right terms. Exo is for chemistry, and ecto, like ECTO, is for biology. So just bear that in mind. Um, and they're a bit different because they relate to pretty much thermodynamics and essentially the flux of temperature and changes in heat in and outwards of a reaction. So that's a bit different, but essentially homeotherms, yes. So Maher got it right. Great job, Maher. And that is regarding homeotherms versus poikilotherms. So the distinguishing factor, ectotherms versus endotherms. I'm just also keeping track of time, but ectotherms and endotherms are under the category of the source or origin of that heat. So think about the heat source. Is it from the body, i.e. in the case of, i.e. of the case of your endotherms, or is it from the environment in the case of ectotherms? Then you have the separate categorization, so it's a bit different, but it's of the rate or the rate of response or the sensitivity to a change in temperature or sensitivity in changing with temperature. So you have homeotherms, which normally keep their body temperature, their core temperature that we're focusing on as constant. So that's where the homeo comes from, which includes ourselves, right? Because we have a narrow range where our temperatures reside in, like plus or minus 5 degrees, usually less than that as well. But then you have the poikilotherms, and they're a different story because they're more sensitive and they thus are able to react and change in temperatures quite dramatically, like the poikilotherms. And so like the Daphnias, poikilotherms, and pot-bellied seahorses that we saw at the zoo, right? So you want to bear in mind the categorization. Even sometimes you may want to consider, based on what you've written down this far, from our Lab 4 assignment, for example, noting the different categorizations of species is very important, right? So if I were to ask you, what is an Arctic wolf? Is it an endotherm or ectotherm? What would be your answer? You guys can share it in the chat as well. Is an Arctic wolf endothermic or ectothermic? Endothermic, right? Perfect. And in what ways is it able to thermoregulate? Think about the features. Think about the external features. Think about the morphology, right? The coloration. This is something that we've talked about before. It makes heat from their body. Not necessarily. That is a minute portion, but it's not necessarily the major factor. Um, from what Meher mentioned to me. But there are a few things. Morphology, coloration. So coloration is the second component. And then the third one is the environmental conditions. So like when we went to the exhibits, right, or the different pavilions, we were able to see different, features of exhibits. And I talked about this yesterday for those that are from Practical 40, that you have like the community, you have like individuals within a single exhibit, right? Like we'll talk about, let's say, the pot-bellied seahorses or the Solomon Island frogs. They're in a specific exhibit. They represent a population because they're the same species living in the same vicinity. And then you have above that, within within a population or above a population, you have communities, right? You can have in some of them like I recall if it's the black monitor. There was a skink. I forgot the name. I'm gonna look at it actually and find out for you guys. But essentially, you may have noticed some of the exhibits have multiple organisms present, right? So think about that. Think about the connections thus. Uh, Methodology is staying in a pack. Yes, that's also another thing Bahar pointed out, actually. The congruence, the dynamics between organisms within a single population. So the Arctic wolves, for example, is a good point. They sometimes cluster together, like the African penguins, right? In the African exhibit, if you guys had an opportunity to look in the pavilion, they like to cluster together as well. That's a third trait. So you have coloration, group congruence or like group dynamics, and their skin colors or their external features as well. Sorry, coloration, morphology, and group dynamics, plus environmental conditions. So those are four main nuances or technical things that you want to bear in mind, especially for understanding the connections between thermal regulation and the categories that those same species are in or fall under. And so I sidetracked, but let me recall. Um, Let me go into briefly a few points. You mentioned poikilotherms or the rates of sensitivity. You mentioned that if they're highly sensitive, they're likely poikilotherms. If they're less sensitive, they're homeotherms. There is like a gradient. I mean, I know we're talking about things like you're either ecto or endo or poikilo or homeo, but reality is that these terms themselves sometimes are variable for these different species. Um, Let me think of an example. Do you guys recall the naked mole rat? It's the only endotherm that is poikilothermic, which has some interesting, you know, connections to essentially the clinical setting, right? Because imagine then you could technically, even though its core temperature uh, relies on or like focuses on metabolism and physiological processes. Just imagine how interesting it is that if you change hypothetically the temperature of the environment, that in turn the metabolism of like the core temperature could also readily fluctuate for this endotherm, meaning that the core processes, the core metabolic processes involved in the naked mole rat could actually change and fluctuate quite rapidly. And that could have some perhaps beneficial clinical use. So that's something to bear in mind. They're not always like endotherms are not always homeotherms, and homeotherms are not always endotherms, and the other way around. But just try to understand that the source and the constancy or the sensitivity to changes in core temperature are things that you want to bear in mind for definitions. And then in practice, the strategies of thermoregulation include what I've mentioned before, the coloration. So the skin tones like the bearded dragon, forgot the name, it was a Suleiman bearded dragon. I'll look to see if I can get access to those specific details. But the idea is that you want to recall the certain features, the certain organisms that we saw across all three sets of organisms. So I'm gonna just open them up and let you guys know, just so that way we can stay on track as well. And if you have questions, let me know. I can't really hear much, but if you guys are able to let me know questions that you have, I could try to discuss them in due time. So in order, technically, we had three different pavilions. Do we all recall what they were? You may want to know this in terms of grouping the different organisms that we were able to see at the different pavilions. You could also look at your manual, page 56. But I prefer if you guys remember it, it'll help make things perhaps have more sense Makes sense. There was the African pavilion, the Australasian pavilion, and the tundra check, right? And so we had different organisms in different locations, right? We had the pygmy hippo, African pavilion. We had the um, Australasian pavilion, the Suleiman bearded dragon. That was the one. That was the one I was talking about. And in terms of coloration, by the way, you notice that for heat sensitive or heat conductive or conducive regions of specific organisms, they may have a darker tone, darker complexion. And that way they're able to readily absorb heat, right? Which is beneficial for like the bearded dragon. It includes also the skinks as well because they're long and they're quite fluid, and they're able to move quite rapidly to different locations. I'm looking at other locations just so I can get it. Yeah, it was a black tree monitor. That was the one. So the black tree monitor was with the skinks, if I'm not mistaken, or was with other organisms, maybe the tortoises. Um, but the point is that, they have differences in skin tone, skin complexion, and that also impacts their ability to essentially adjust to changes in temperature. And the black tree monitor was also ectothermic, which is another point to bear in mind. Um, the second point, now that, like, imagine you have the skin, right? Something to compare with is external features like the fur, right? Like the Arctic wolves or the polar bears, right? So for the polar bears, they have blubber, right? 11 centimeters thick. It's like a thick layer of fat, essentially, which even though it's internal, it connects to the thermal regulation as endotherms because they can metabolically absorb, conserve, and break down the fat when they need to, especially if they're like in mode for hibernation or hiatus, essentially of trying to conserve and make use of energy that is internal to sustain themselves, and essentially, um, you know, follow suit with homeostasis. So think about those different connections, and also polar bears are homeotherms, and the same for Arctic wolves. Even though they live in colder environments or climates, they still exhibit these similar traits that we as human beings have as well. So you want to think about those details. And then that's the second component, right? Like external, like you have fur as another component, as I mentioned. And then you have like um, what was the other component? Morphology, right? So morphology, that includes, the fur, that's one component. You have appendages, right? You had like the yabbies, the red-clawed yabbies that essentially have these pseudopods as they swim, to essentially move in multiple directions. And similar to actually the gills of the pot-bellied seahorses, they are able to move in multiple directions, three-dimensionally. So they can go forward and back, left and right, up and down. So those are ways for different organisms to move and migrate to different locations, which actually connects us to our fifth lab. We're connecting to isopods, right? Because when you recall about chemotaxis or even taxis in general, it's a level of attraction and mobility from one location to another, right? So depending on what the stimulus is. So you want to think about those connections, like how I just have shown you, in terms of like morphology or mobility, and how those two have interestingly connected labs 4 and 5. And then that's something that I really wanted to touch upon. And I think I hopefully have mentioned to hopefully come to let you know the importance of integration in our labs together. Um, the third component when it comes to thermoregulation, though, is like the environmental strategies, right? Or the environmental conditions. So let's say, some organisms live in outside, like open exhibits, right? So you had like the American bald eagle, and the Arctic wolves, you even had the Omer tigers, right? You had certain organisms that would be exposed in the open environment, so it wasn't really controlled, right? You can't really control for humidity, I mean, unless you have it enclosed really for temperature and other conditions like exposure, etc. So you want to think about those perhaps limitations, just like how we control for the setting for the isopods themselves, right? So you can really hopefully come to appreciate the connections in this regard. And indeed, they're interconnected and interwoven in unique ways. Um, let's see, what else? Let's see. So environmental conditions, yes. So even let's say going back to the Black Tree Monitor, this was in, I believe, the Australasian exhibit or pavilion. It's one of the most arid continents, so essentially connecting Australia to Asia, the species that we were able to see. Some of them will take measures to essentially bask, right? Like think about tanning under the sun. Like if you go outside right now, you'll notice the sun is quite beaming. Uh, if you're around the student center, for example. But then others will evaporate and sweat. Like so they'll do evaporative cooling, like the Solomon Island frogs, right? So they'll essentially sweat tears. Even similarly, the pygmy hippos or the crocodiles, right? They're able to sweat tears. But for the pygmy hippos, they actually have a blood sweat like antioxidant surface. So they essentially are able to better regulate their temperature or essentially thermoregulate by cooling down, but also removing waste products through their blood secretions. So it's a unique way to also purify their blood, right? Because even if you think about yourselves, imagine hemolysis. This is a separate point, but you have red blood cells that get renewed, and that they they regrow accordingly or they redevelop. You have some that grow and some that die. And so you have like debris that does exist in your circulatory tracts, right? And that's something to also bear in mind, connecting to your module two for Stephen Reed for physiology. So you want to think about those components, those connections indeed. And that's something I really want to integrate. But when you're basking, right, that's a way for you to essentially increase in core temperatures if you're, let's say, an ectotherm, like the green tree python or the emerald tree boa, right? So those two, having a dark, like a green complexion and one having stripes, one having dots. I believe the dots was the emerald tree boa and the green tree python was the one with stripes. But the idea is that, they bask and they may migrate, right? So they may move up and down to different locations, places that there are shades versus places that are more exposed to ceramic heat lamps where heat can be absorbed or adsorbed. So think about that. Think about also the exhibits, right? They're able to essentially modulate those different conditions. Any questions, by the way? I know it may be in a bit quick, but I want to go to, I don't want to lose time for you guys to go to uh, week 5 material. And if you have questions, we could go, let me just actually share the screen. But nonetheless, like you guys can think about what I've mentioned, think about the implications, the integrations, and the potential for investigation or the integration, the integration, investigation, implications, or the implications really with the thoughts that I've described thus far. I'm going to share the screen. Also, if anyone's speaking, please let me know because I can't really hear. So now you're gonna see me with you in slide. Right, so it's very interesting. Hopefully you guys can see me and or the screen. Can I get some emojis, thumbs ups, if that is the case indeed? Yeah, okay, I see Meher. Yiping, Joshua, Jeffrey, um, Dorlin, all of you can see the screen? Yeah, okay. So let me know once again, if you have any questions, please feel free to, we'll save them maybe for the end, but I just want to make sure you're all paying attention. Very important indeed. Um, Let me just go straight ahead now. Okay. So we had, our pre-lab, the agenda items. We looked through population growth, right? So this was the beginning of our Lemna experiment about 3 weeks ago. And so hopefully, like after you've looked down into, and what I recommend is looking back at your manual to see more details. But you have duckweeds, right? There's specific introductory material that you want to bear in mind, especially bolded items or items that take or like terms that take a bit of time to digest and understand. And when you're preparing for your lab exam, bear in mind that you need to know the content, the introductory terms, as I mentioned for homeotherms, the poikilotherms, and etc. Um, endo-ectotherms included, and understanding the implications. That's like you have your methods, limitations from them that exist, and the controlled variables, things that you wanted to measure, different parameters from shoot length and shoot biomass from weeks 1 to 2, and then like looking at heart rate from week 3 or week 2. But nonetheless, describing these different components that we've hopefully gone over, and then connecting to your discussion and results actually results in being able to articulate well the different parameters that you've explored, interpreting the data thereafter, but making sure that you're also very familiar with, stats, the different, formulas that we've been using from your lab manual. Bear in mind as well, like the figure captions, table captions as well. Specificity is needed, right? When you're making a figure caption, it should include an intro to the aim or the figure itself or table, what it's representing, or the parameters, the species, the variables at play, the assay as well, or if possible. The second sentence is normally referring to the brief mention of your methods and different parameters, conditions that you were, adjusting or manipulating until you reached that result, right? So what you did to get to where you were or where you are now. And then the final set of sentences includes a summary of your results. And that's something that I want you to think about. Some labs will also ask a third or final sentence describing some interpretation, but overall, the three sets of details of your intro, methods, and results is what I normally require. It helps also, students that are, even as we speak, completing their FLRs for a course I'm marking or a course I'm TAing as well, called microbiology for C17. So just bear these details in mind. They're fundamental and pivotal for not just this lab, or even perhaps your lab exam, but importantly for your other practicals in the future from different courses that you may take. So think about those details as I describe them. And moving onwards to, the setup, as you're aware, we are able to study different concentrations of NaCl, right, and their impact on the growth of lemna. And how we're able to do this was measuring the different, the initial to final number of thalli in total, right? So we had 4 different lemnas, 3 thallis each. That would give us 12 in total. And so essentially your initial, as we recall from our table that you likely would have completed for your Lab 6 assignment, you would have essentially counted the initial and compared it with 14 days after Lab 6 material, your results thereafter. So you would essentially account for the different conditions. Remember how you were able to calculate it, right? We used C1 times V1, i.e. the concentration of your stock multiplied by the volume that you're using, typically 20 mils, equals to essentially the final concentration of your diluted treatment of NaCl, which is shown, I believe, here, multiplied by the volume in total, which would be with the artificial pond water, a total of 200 mils. So generally speaking, your stock concentrations would be 10 times larger because you went from 20 mils to 200 mils, so you're diluting by a factor of 10, meaning that your concentration to start off with would have been 10 times larger. So if you look at all of these conditions from treatments 1 to 5, you would have noticed that your initial stock concentrations of NaCl would have been 10 times larger. So bear those details in mind. Inshallah, I want to make sure that you guys understand these concepts and that you were dealing with these in reps, right? So you could see the formula. And let me know, folks, if you can hear me. I apologize. It's a bit loud, but nonetheless, what I will say is that have a look at the table that you see also in your lab manual, and I hope I gave you guys a good understanding as to how you would get 20 mils here, and to get essentially 180, because 200 minus 20, 200 being the final volume minus 20 as your stock would have given you 180 mils across the board for artificial pond water. So bear those details in mind. It mimics using artificial pond water rather than DH2O or doubly deionized water. It mimics the aquatic environment, so the aquatic bioassay, which reminds me of our thermoregulation exercise with the fishes at the Australasian pavilion, right? Even if you had a moment to look at the Indo-Malaysian pavilion, I actually went there thereafter. So if you want to look at the recording, you can see it also on the server. But the idea is that you could be able to come and appreciate the different, organisms, especially those under the ocean that are highly impacted by pollutants, which connects us to Lab 6 material with the use of NaCl as a deposit and its impacts on aquatic growth, which is, by the way, unique in connecting actually this material to BIOC17. Because essentially, for those that are interested in microbiology, we'll deal with bacteria that are also in unique environmental conditions. That includes changes in fact, salinity or osmotic pressure, which is done through increases in NaCl content. So students actually in that lab that I was teaching last week and even this week were able to see their results following 7.5% growth, 7.5% incubation of NaCl in specific plates known as differential plates. So they were able to see certain bacteria grow and some other bacteria as well. So inshallah. So sorry. Just bear those details in mind. Try to connect the dots. That's my recommendation. But as you go forward, hopefully you'll be able to appreciate the connections in turn. And then using the dissecting microscope is where we, started to look back into the past. We used it before with the Daphnia. We used it for the celery the plant cross sections using the microtome. And now we're using it in terms of, observing the morphology of isopods, right? So we had the Porcilio SPP subspecies. Hopefully we're able to capture the morphology, the appendages, the uropod, the legs, the eye, the compound eye, which is similar to what you saw from Daphnia as well. So bear in mind the trends, the similarities, the differences as well, right? Because those different components are readily connectable and integrative to what we explored at the zoo as well, where we saw different types of organisms, maybe 30 of them across 3 different pavilions, if not more. And, just come to appreciate that, independent of the species, some have fur, some have scales, some have gills, some have feathers, yet they uniquely, subhanAllah, have similar trends, modes of thermoregulation that you really want to consider. And now when you want to investigate, right, this was a pinnacle point and tenet or principle I talked about in the past in the I-5 approach. Investigation was one of those I's. And so you want to think critically, understanding that you have independent and dependent variables and even controlled variables that you want to bear in mind. So think about, like as we did for the independent variable of this exercise, following your analysis of the isopods and their morphology. We want to think about, and by the way, these isopods have gills, by the way. It's quite interesting. Even though they're terrestrial isopods, they also have gills, external gills. So quite interesting in the respiratory system as well. But nonetheless, independent variable, what were you examining, right? You were examining you were analyzing different substrates for different groups as stimuli, and then thereafter the response served as a dependent variable. So your independent variable focused on certain stimuli. Sorry, give me one moment. So I may have to use the washroom. I'm a bit tight with time, but let me know if that's okay. I hope you guys don't mind. I'm just gonna pause. I hope you guys don't mind. Hopefully you guys are able to pay heed in due time. Let's see where we are. Okay, so we have about 15 minutes. Let me just go over the material. So, when we think about like this experiment, at least in your hypothesis, remember you have an alternative, and so within your research hypothesis, it's usually broken down to separate an alternative from a null hypothesis. Your HA refers to your alternative, and H naught or HO refers to your null. Some people use HI as well for alternative hypothesis, but the idea is that once again, you have a hypothesis, you're making an educated guess per se, really trying to describe. Um, sorry, give me just a moment, folks. Um, you have your hypothesis, right? And you're trying to observe how your independent variable impacts your dependent, right? So in this case, we had different substrates, different stimuli across different categories for like chemical, sugar, and food sources or stimuli. And the idea is that each of those different categories, from chemical to sugar to food, had different categories that could be explored as independent variables, right? So you could have had within your independent variable of chemical stimuli, the use of glucose, acetic acids, sodium chloride, and sodium itself. Um, and something that you want to bear in mind, like if you know a bit about chemistry or even biochemistry, you would notice that acetic acid, as you are probably aware, is an acid, right? So you don't typically you wouldn't expect, especially insects, to be sent to adore certain acidic environments. So you could likely expect that it would more favor towards a glucose or certain metabolites that you could ingest. And similarly for sugars, it may want to absorb specific monosaccharides like glucose, galactose, etc. And then food sources, think about like what type of food source isopods may like to endure, right? So that being said, you weren't tasked to especially predict which of the following categories within your stimuli would isopods best favor or react towards. But nonetheless, you want to get an idea that you have an independent variable, and within that, you have different categories that you could explore, right? So your independent variable could be chemical stimuli, and then the different categories that are categorical as shown here could be provided for glucose with its concentration and acetic acids, sodium chloride, and sodium in, I believe, weights per volume, which is just a nuance, but just something to bear in mind for units. And then you have the sugar sources, the food sources, very similar indeed. But then your dependent variable and how your independent, i.e. the stimuli, relate to your dependent variable reflects the response, the taxis of your isopods. And that numerically is quantified by the number of isopods per well, right? So you want to think about those four different categories as you see per independent variable or per stimuli. You can notice how we had four different wells on the outer regions for that purpose, right? And so that's where you would likely want to see how the isopods move and migrate to specific stimuli and if they deter from specific ones as well, right? So, and that's where you would have quantified after 15 minutes the total number or the number of isopods in specific wells that you were able to essentially inoculate with filter paper absorbing those specific stimuli or categories within specific stimuli. So just bear that in mind. It's more like the conditions or the substrates per stimuli that you're inoculating in each well. But the idea is that you would have done this and hopefully were able to measure the different effects accordingly based on the number of isopods per well. And then what you would have controlled for, and bear in mind the natural environment, the setting of these isopods, right? They prefer humid, moist, and dark regions or locations. So if let's say they were in the Toronto Zoo, they'd probably buried in a small barrel in a humid, like the exhibit would probably have a humid controlled center, so that way, they could stay and migrate and grow however, but in optimal conditions. But the idea is that you would control for, as we're in the lab, for specific, sorry, Um, you would correct for and control specific settings or consider specific variables that you would like to control, like humidity, light exposure, water abundance, temperature, etc. So these are things that you want to bear in mind. And then even as a control for both control and experimental trays, you made sure that you would have added just water into the middle tray for both. But for your experimental, you would have had all of the four different stimuli did it say stimuli or four different substrates per stimuli in one of those other wells, in one of the outer wells, and all four of them. And then you would have had for your control all five wells just with water absorbed onto the filter paper. So that's where the two different trays come in, because you would have wanted to quantify as you were doing the chi-squared analysis the impact and the essentially association of isopods to specific wells and how that fares well with what you would have expected based on your control. So you have your experimental trial, right? After letting them remain moist in this arid environment for about 5 minutes, letting them acclimate, onto the filter paper. You would have let go of the middle physical plate, and the idea is that they would migrate, right? Every minute you would keep counting until you reach 15 minutes. And they would have started off as 10 isopods likely at the middle, and then they would have migrated accordingly. You may have also observed their own interactions with one another, but the idea is that you would have nonetheless tabulated, quantitatively the different number of isopods per well. And inshallah, you would have also compared those results with the control, right? So the control had water all across the board, and the benefit of that is that you were able to thus compare with what you would have expected, because technically speaking, if you have 10 isopods in 5 wells, you should, at the end of the day, roughly get 2 isopods in every well, including the center, right? So just bear those details in mind. And that's where you want to think about like this research question and how you can make and develop a hypothesis based on what you would have predicted. And so after that, after you've grabbed, I'm looking now into pages 70 and 72, you would have, essentially placed your well numbers, the categories of the specific stimuli, like for each of them per stimuli, like if it's food, chemical, or sugar. And essentially, the observed number, frequency of isopods would have been placed in the second column, and then thereafter the expected number would have been 2, i.e. O would be variable, something in your last row from the larger table of pages 69 and 71. But the idea is that you would have thereafter placed E, which would have been number 2, across the board for the third column in tables 5-3 and 5-5 for your control and experimental trials, respectively. For your last column, you would have used the formula to essentially determine the number of observed, to compare the number of observed isopods per well, subtracted by the number of, subtracted by 2, which is the expected number of isopods per well. And then you would have divided that, or you would have squared that value by 2, and then divide that by expected number, which is also 2. So a bit of 2's around the board, but it would have been something that would be quantifiable for each of the wells. And thereafter, you would agglomerate, or you would accumulate and essentially add up all of those different terms within the function shown at the top, which is like essentially from the fourth column, you would have added all of the results or the solutions for each of the wells and categories to what would have made your chi-square calculated value, right? Which is the sum essentially of the observed minus expected squared divided by the expected ratio. So you would have summed out all of the those mini chi-squares into the chi-square calculated value. And I want to make sure you guys understand that because you are calculating, and in order to compare your results to compare the observed with what you would have expected, you need to bear in mind the chi-squared calculated versus critical values. And so essentially that will connect us to the next slide, but just once again, null hypothesis versus alternative. The null assumes that there's no significant difference between your observed and expected data, i.e. between O and E, whereas your alternative hypothesis indeed suggests a significant difference between the two data variables. So bear those details in mind. If you had 12 isopods, that's why you would have 2.4, but in our case we had 10. And then I just mentioned about the chi-square calculated, but your degrees of freedom would be 5 wells minus 1. So that's essentially 5 categories minus 1. 5 minus 1 is 4. And so based on, this is very important, understanding where you get your critical value, your chi-square critical value from. You have your degrees of freedom. I mentioned that's 4. But then you're going with your alpha. And I don't know if you learned this in biology, but alpha is referred to like in stats as your essentially probability. So think of that as your p-value, which is essentially in this case synonymous at 0.05. And so you want these two lines to intersect, which is where you would have gotten essentially this value here as 9.488 as your chi-squared critical value. And I just want you to understand this because you need to now compare your critical to your calculated value. When you're doing this, you'll get a better understanding as to what your stats conclude. And essentially, if you have your calculated value smaller than your critical, then you fail to reject the null hypothesis, i.e. you accept it or you assume it. Whereas if it's greater than, if your calculated value, let's say is like 10 or 9.49, and it's greater than your critical, which we know here is 9.488, then you reject the null hypothesis and you assume the alternative, right? i.e. that there could be a significant difference between your observed and expected data. And then so that's where your chi-squared values come in. P-values is where you normally do your t-tests, right? And so generally when you have your p-values, as you're aware, if it's less than 0.05 or equal to that number, then you reject the null hypothesis. And if it's greater than that, then you fail to reject it. Does that make sense, by the way, everyone? Please give thumbs ups or reactions if you find otherwise. Okay, I see one from Meher. How about everyone else? Jeffrey, Joshua, I see a thumbs up. Jeffrey, Dorlin, Yiping, you guys understand? Okay, perfect. Okay, so let's move ahead. So I'm just needing to move a few things, but okay. So we've talked about this now, and then that's where your lab 5 assignment comes in, right? You would have plotted the data, you would have shown your stats, shown the table, would have used a table caption with a clear description of the intro, methods, and the results briefly. And then you would have had specific questions about if you rejected or failed to reject either a hypothesis or both, then what would you have concluded? And that's where my, I5 approach comes in because you have the implications there on discussed, right? So what are the implications of your results? And if you think about your FLRs, think about like the extensions, the implications, the limitations that you would have observed from this exercise and future ones as well. That's how you begin to think critically, folks. So that way, I know many of you are, but I want to reiterate or regurgitate this because it's very important that you understand the concepts and how you can critically articulate the different results that you have observed across the different practicals, across the exercises, and across even time that you've been in the practical with me. So think about these different questions. Hopefully that summarizes a bit on note for our last exercise or semi-last one. Any questions? Let me actually go quickly to the next one. Sorry, folks. Oh, My computer froze, so give me just a moment. I'm sorry, I think I missed some things in the chat. Okay. So give me a moment. Okay, no questions. Perfect. Where can you find the slides? Okay. Um, Technically, I can't give you guys slides. It's a bit, I know this may be hard to digest, but technically, I'm not allowed to give slides to students. So I just want you guys to at least understand the material. And if you have questions, follow through with what you've seen from the manual. But if you have questions and you want to ask them to me, I'll be more than happy to refer to your questions accordingly. Right, so I may actually pause for a moment. I'll go use the washroom and I'll join you guys shortly. I wasn't able to use it before, so I'll come back. Hopefully, I'll see you guys shortly. Hopefully that's okay. So just stay tight for just a bit or some time. I really appreciate it. Okay, folks, I'm back now. It's a bit difficult to use the washroom actually, so I'm just waiting. Or let's see. I think I may get an availability soon. In the meantime, let's go briefly, or let's continue. No, okay, to reshare the screen. Okay, so we're back now, and as you can see, we're looking at the lab exam. So this will be a good idea, inshallah ta'ala, to actually remind yourselves about the different concepts, about like understanding what you need to expect, and the format as well for the lab exam tomorrow, 2 to 4 p.m. Yeah, 1 hour once again, but it's synchronous, in synchrony, in the sense that you have only 1 hour to do it. So try to budget your time when you begin. I'd recommend beginning like maybe 2:51 maybe, so that way you have at least 50 minutes to just go over the material and then a minute to start on your computer to accordingly begin the lab exam. But I've already touched upon, that we're now in the lab 6 with like the final lab for review. But I want you guys to understand like the concepts of what I've described, integrating the components between exercises, between practical weeks, and hopefully with outstanding questions that may connect with novel situations in your exam. So you want to understand, integrating, investigating, implicating, and integrating the concepts in unique ways from what I've described thus far and beyond. Okay, so any questions, by the way, regarding the lab exam at least? Just before I go back to, for those that are in Practical 40, this will make it easier because it's literally things that we talked about yesterday. Yeah? Okay, no questions? Okay, perfect. So by the way, separate point, but if you could please scan the QR code, I'd really appreciate it. It'll be beneficial, inshallah ta'ala, for me to know how to be a better TA. So if there's anything that you guys want to share, any compliments, considerations, concerns, anything that you want to share, please let me know, or feel free to share it in the anonymous form here. I'm just gonna go down. So we, following lab 5, just like lab 1, we went 2 weeks later to look at winter wheat into lab 2, but now into week 6, the growth of your lemna, right? The duckweeds. So you want to now, move a few pages further. And if you look like back into the past, you'll see some, formulas from pages 70 to 75. But then into lab 6, you're looking at material that focuses on lab 6B, which is essentially the material regarding the question, is road salt harming our aquatic system, our aquatic ecosystems? And think about systems in that way as well, because the zoo, as we talked about the Toronto Zoo, is an example of an ecosystem. You have communities within pavilions, and you have populations within exhibits, right? So you want to think about going up and down, left and right, forward and back, the different implications of our exercises together, right? So think about those details that I've described thus far. And let me just see, I'm looking at yeah, you'll actually even find it quite abundantly connecting to other courses as well. Like as I've just mentioned about C17, a microbiology course in third year. So think about these different connections as you will see shortly. We talked about the effects of road salt, right? Talking about the environmental impact, the impact of excessive road salt use nowadays, or like essentially you want to think about like, the impact of these deposits on our roads, on our sidewalks, because plants still grow there, right? And then on the separate point, when you have those deposits leak through into water streams, into reserves, into seawater, then you can just imagine how drastic the impact is, because it's not simply, going down a stream and liquefying or essentially solubilizing, but also just imagine how many other streams interconnect to that stream that now is becoming a river filled with road salt. And then just imagine how that gets compounded, not just here in Scarborough or in Pickering or Whitby or in Mississauga, but or even in Etobicoke, but all of those different locations now following, rain, with rainstorms and floods, etc. Imagine all of those, deposits all going into the 7 oceans that we have around the world, right? So just think about the impacts of us on many aquatic organisms, as we've spoken before at the zoo and beyond, right? Even our Daphnia in the lab to, the isopods, right? The terrestrial, but, will like a sip of water when they can, right? So think about like the importance of the implications of, different deposits, some of them being toxic. And even we talked about this at the zoo with the Great Barrier Reef, the coral reefs, the different aquatic organisms that live surround the reefs, and even the reefs themselves as ectotherms, right? So think about the impacts accordingly of different organisms and their environments and the exhibits that surround them, and the different organisms that interact with them in their different functions and interactions around the world. So functions, forms, and interactions is the course code, or the title for our, our course. But nonetheless, let's go forward. I hope, by the way, many of you understood maybe lab 6 for exercise 3. I know we didn't do it in person, but hopefully you got to see the results and could understand the material from what was also mentioned. Let me just see. In page technically begins lab 6B from page 85 all the way to page 89 to nearly 90, halfway into 90. So you want to understand that essentially carrying capacity is where like, the uppermost limit is reached for organisms to grow. And I was about to say bacteria because we deal with bacteria and microbiology. And so it's quite interconnected, these results that you're looking at in logarithmic form. So just think about, or in the exponential phase, which bacteria also go through like E. coli. So think about these different details, connect the dots accordingly. And then for exercise 4, we looked at the lemna themselves, right? Following 2 weeks of growth. Sorry. So we were looking, remember that we're counting the thalli. So it was a proxy, i.e. like a mode of measurement to determine plant development over time, right? So after 14 days, they would have grown with any of the 6 different treatments, 1 control with no salt included, and then the 5 different increments of concentration increasing in double from 0.625 to 1.25 to 2.5 to 5 and 10 grams per mil. So you want to think about, sorry, is it grams per mil or grams per liter? Maybe grams per liter, sorry. But the idea is that, you want to bear in mind these details as you compare the different conditions, the different number of thalli that were able to grow, and correspondingly observe the impacts of growth rates on an absolute level, i.e. like, I'm actually going to show you guys the comparison here. Let me just see if it's, yeah, so this is practical 39, but the idea is similar to what you guys would have expected, right? So you would have your initial starting number of thalli, moving to the final set that we saw yesterday if you're from practical 40. And so you would see like in the case of practical 39, they had almost double many of you guys in the number of thalli. It's not a problem. Like, don't feel concerned if yours was like half that size, even at the control, because essentially following the logarithms or the natural logarithmic calculations, I don't want you to just look at this. Yes, there is a difference indeed that is quite comparable or incomparable because logarithms go by a fold of 10. In this case, natural logarithms by at least 2.618. So nonetheless, there is a difference. But the idea is that you would have essentially, as you recall, use this formula to determine the growth rate for each of the different reps, and then thereafter determine the mean, which essentially is the average of those 3 reps per treatment group. And then thereafter, you would have repeated the steps for the ones shown here. And so you would have just copied and pasted the formula, or you would have just essentially took the average. You would have taken the average for the reps as shown. And then importantly, your growth rate is once again relative to the control, right? So the control relative to control is 100%. But when you're doing these calculations of the latter treatment groups, you need to bear in mind that the percentage is your mean growth rate for that experimental group, shown like in the same row, divided by the control. So you would have done 0.209 divided by 0.330, and that would have been, inshallah, multiplied or converted with the percentage format, right? So I normally adjust the percentage because it'll give you in decimal form. So that way you could abstractly see the changes over time or over changes and increases in NaCl or sodium concentration, sodium chloride concentration, right? So you just want to think about these details accordingly, right? Because I mean, you saw these trends here that many of you would have likely seen a decrease over time, right? So I'm just going to show like abstractly an example of this. So, and don't worry that you don't actually, let me show it definitively. So let's say I was going to take the final average. Like essentially it goes down to maybe 50% or less, roughly, in value. But you don't have to worry about too much. Like generally speaking, so you see here is like 49%. I don't want you to worry too much about it, but I want you to think more abstractly that even when, you account for these mean growth rates. Percentage-wise, even though we have group 39 and group 40, practical numbers, having maybe double the number of phthalate at the beginning for the control, and double the number of phthalate across the different treatments. Something that you want to bear in mind is that when you look at your own control as your practical, you reflect back to the percentage growth rate. And generally speaking, even though it was double here, right, or double in the previous practical, you would come to appreciate that the trends in percentage growth remain relatively similar, because once again, you're referring to your own control, and then your own control is half that amount. And so independently, you are still considering, and you're pretty much almost pseudo-controlling for the fact that there may be half that number of phthalate across the treatments. So the percentage growth rate doesn't change drastically. So that's where you would normally would have seen, as you increase the number, increase the concentration of your sodium chloride, you would have seen thereby a decrease in growth of the number of phthalate in your Lemna aquatic duckweed plants. So think about those details. That really pretty much, I hope, summarizes a good chunk. I think there's still a bit that I could describe for you guys. Go back down. Yeah, so subhanAllah, we talked about these populations, right? We talked about the definitions and communities, and that's where you connect back to conservation efforts. Yesterday, I briefly talked about this, but birth and immigration. Do we know the definitions, everyone? Like birth, you're having more, offspring, progeny produced into a given population within that population, so it increases in size. Immigration is like you essentially having new species or individuals come into your population, so thereby you're increasing also in turn your total population size. Emigration is the opposite. You have, species or individuals leaving that population, so it decreases in size and mortality, that you have organisms that pass away, and then you have the population decreasing also in size. So you want to just think about those stats, understanding where the impacts can exist, because if you have, for example, more births and immigration than emigration and mortality, then that could be a good sign for the growth and successful conservation, or I would say the populational growth of these organisms, right? That connects us with the phthalate, that connects us with the isopods, maybe not the isopods, but the winter wheat, for example, right? So think about the sensitivity of certain organisms to be able to grow and to survive. And that's actually something that we've seen quite abundantly at the zoo, because as you can imagine, if there is a decline or the net birth and immigration rate is less than the net emigration or mortality rates combined, then you may have more species leaving your population versus more that will come in. And then thereby the population size would decrease because you have more mortality or emigration than birth and immigration. So think about those conservation efforts that then come into play, like for the American bison, the Solomon Island frogs, for the Great Barrier Reef, right, and the many organisms, aquatic organisms that live in this environment, this auspicious, this unique environment indeed. So think about the implications thereby, and how thereby you have conservation efforts in play, and that's where we're getting into the microcosm, because you have us now looking at beans and rice, for example, right? So the mark-recapture method, right? And that's something that you want to think about, because, unlike quadrat sampling, you're not going through a grid and estimating the number of populations or number of individuals within a specific grid and then multiplying and averaging out to determine the total population size. And in this case, you're taking a different approach, which is even, believe it or not, done here in Canada, right? You may have seen you see this red cardinal, right? It has a tag right here. Or I don't want to go on tangent, but there are different unique ways for market capture to be done. And you guys could actually search it. You'll come to appreciate that what we did was actually a legitimate exercise that actually could be recapitulated in the real world. So, and biologists and ecologists in particular do this quite often. So as you recall, we had three different sets of experiments, mini experiments, in which you marked 20 beans. You would have placed them all together with unmarked beans into a container of rice, mixed them, shaped them up, and then you would have taken out 20 random beans with with cups essentially, right? So and then you would have returned the rice, by the way, each time to mimic the constancy or to control the amount of rice present in the containers. Otherwise, you would essentially be uh, concentrating the number of beans as you would essentially take out more. So you would likely take out more and more if you were to reduce the number of rice or the amount of rice thereafter. Um, so just bear that in mind, like you want to make things conformed or um, you want to maintain the original density per se. Nonetheless, so you are taking out beans, so that does reduce the total population of beans inside, right? But the idea is that you would have done this, you would have counted therefore uh, M1 being 20, C being the number of total captured. It's not technically recaptured, but total captured. Um, oh, total captured beans, right? It's a bit slight of something that I want to clarify because you're capturing the first set of times beans that are unmarked or marked from the rice container, right? So you would have likely gotten something around 20, 18 to 22 is also okay, but around 20 is ideal. And then looking at the ratio now of R. R refers to the number of now marked beans out of the 20 that you would have captured, or out of the C number of beans that you would have captured. And you would have done this with experiment 2, just repeated with taking out another 20 beans. So you would have still kept those 20 outside of the box or container. And you would have repeated this process with 20 more beans now outside. So hopefully you would have seen a greater number of marked beans, or even just a greater number of beans to derive for M, which still would have been 20, but C, which would have roughly be 40, and then R, which would likely be greater than M1 or R1. So R2 would have likely been a bit greater proportionally. But then, or not like essentially a minor increase if possible. But then you have experiment 3, which is essentially now you using all those beads that you've now, sorry, not all beads that you've captured, but just marking 20 of the beans that you have that are unmarked, either outside or inside your container. And then likely you would have done the unmarked beans. So out of the 40 that you now have outside, you would have essentially just marked 20 more. So that way, in total, in your container, including in this entire system, you would have gotten 40 marked beads. So that would have been exercise 3 with M3 now 30, M3 now 40 roughly. And then you would have reset the process of continuing experiment 2, now taking out 40. So using the cups once again, and essentially capturing 40 beans and then determining with R the total number of marked beans. So with that, you would have gotten 3 different types of values for M, C, and R across the different experimental methods. The idea is that you would have used a formula, which I hope you guys are able to understand. I believe it's one of the final formulas that we learned. But it's referred to in page 79. If you want to look at your lab manual. So essentially, R is equal to, don't know if we have a formula on the slide here. So I'll just mention it. So you have R divided by, I think I should actually write it here to make it easier. Let's see if I could pen it out. So you can use R. R divided by C So think about proportions now. These R refers to the number of marked beans. C refers to the total number of beans that you captured. R being the ones that you've marked. And then proportionally, you expect that the number of marked beans in total, inside and outside, should be proportional to the division of M by N, capital N, referring to your estimate. Right? And so if you wanted to determine N, you would have essentially moved it to the top, and that would have been, and then you would have flipped R and C to be on the right-hand side. So you would have gotten N equals M times C divided by R, meaning that the estimate of your total population size of number of beans in the container filled with rice would be essentially your total number of captured beans multiplied the total number of marked beans, like total in an entire set of containers, divide in your container, I mean, divided by the R, which is essentially the total marked yet recaptured beans. So just bear those details in mind. Try to understand, like, the crossover, substituting or the essentially isolating of the variable n, so that way you're able to understand how you can complete these specific questions if asked upon in your exam. So I'm just gonna go forward. Does that make sense, by the way? Everyone? Please give a thumbs up if that makes sense. Hey, Maher, great job. Great job, Maher. And Joshua, I think that's also you. Sorry, I can't really see the screen. Okay, Dorlan, was it Joshua also gave a thumbs up? I couldn't see. See Dorlan, Jeffrey Yiping, and the person with the period. I don't know who the person with the period is, but feel free to let me know accordingly as well. Perfect. Okay. Jeffrey as well. Okay. I know you're paying attention, both of you. Well, all of you, hopefully. And then Yiping as well. But inshallah, let's go forward. So I'm worried if actually, I think I have to edit something. Does that make sense, the formula, by the way? Okay. Let's go forward. Yeah, that's why I was wondering if the annotation would still come in. Okay. So I'm just going to clear it up. If you have questions, yeah. Okay, so let's go forward now. So I want you guys to actually look back for some of us relatively recently, look into pages 79 and 80 again, because yes, I provided the formula for n, but you want to also do a similar, like you want to derive essentially with the variables that you have for MC and R, the standard deviation and the variance. The relationship between variance and standard deviation is that variance is squared the value of standard deviation. And so if you want to determine standard deviation, you'll take the root, the square root of a variance. So you see the formulas on the end of page 79, and then the confidence intervals are determined as not this setup per se, but actually, if you look carefully, it's essentially n plus or minus 1.96 times your standard deviation. So just bear that in mind. That is n plus or minus 1.96 times standard deviation. That's the top of page 80. And the reason why this is important to understand is because you thereby, if you have the plus or minus, you are accounting for a plus end or an upper limit to a minus end or a lower limit. And that reflects your boundaries or your confidence interval indeed. So how confident you are that the true estimate of your population, i.e. lowercase n, is within your intervals. Or essentially you could consider it as like you are 95% confident that n, lowercase n, is within your calculation using your estimated n, uppercase n, right? So think about the connections in that way. Like you're kind of assuming that lowercase n is within this framework of where you calculated uppercase n. So just determine or use those values accordingly. I'm just going to correct something. Yeah, so you use essentially the uppercase n to determine your confidence interval, which would reflect how confident you are that lowercase n is within that interval, right? So, and that's something that you would have seen to compare across the different methods, and I hope you guys have been able to do this, because technically your error bars should be equal. Upper limit and lower limit should be the same distance from your estimated uppercase n, estimate of the population size of beans within your technically vial. So you would have essentially done this and it would have compared them across the experimental methods and then would have tabulated or graphed the number of individuals that you see here for beans essentially. And then you would have plotted based on what you know from lowercase n, the total number of beans that you would have calculated or counted from the very beginning or end of the practical, either yesterday or last week, Wednesday. Yeah, so with that, any questions, by the way? Okay. Good question. Will we get feedback for our lab 6 assignments today? That's a great question. Okay. I don't think you can technically. A few reasons. One is that Sejani, the course coordinator, would have to, like, even if I'm done hypothetically marking, and like I'll actually have FLRs to also mark. But let's say hypothetically I was done, you would still have to wait for Sejani to publish or to upload your grades, and so you may not get access actually. I don't know if you get access even today. So that's why I would recommend actually asking me questions, or if there's any like um, pressing concerns that you want me to go over, then I'll be happy to go over those questions. But do bear in mind that you're not going to have too much content on Lab 6 because after all, many of you would have just done it yesterday or this past week. Some of you would have even done it today for those on Thursday tutorials. So just bear that in mind. That's a great question. FLRs, I'll have to see. As of now, I can't provide a specific percentage because I have to look back and I'll be honest with you, I'm a bit like a bit fixated with time. But at the same time, if there is a change in grades, like if your grades are scaled, then I'll let you know. On Purkis, inshallah. That no problem, no problem. More than happy to help. I have scaled though labs 1 to 3 for assignments because I wanted to make sure I'm fair in that regard. But we'll see how the other ones fare. Let me just see. So cleanup, population method, we talked about this. And the lab exam, we're returning back full circle. And you would have also saw this for the review from weeks 1 to 3. So if you guys want to have a look, sorry. If you have questions at least, or if you want to look back into your manual, look back into my recording from the past, you would have to email me, but just let me know. And I also have posted it online as well. I could post it on Quirkus, but I would prefer that you guys, everyone that wants to attend pays close heed and benefit to what I've mentioned thus far. Any final questions? So are we allowed? Okay, that's a great question. For notes, see, I think you would have to ask Sejani. I don't know so I haven't been given details regarding what you are allowed and what you're not allowed. I don't want to like open, like, because if I open my window, you may see confidential stuff. So What I would say is ask Sajani. To my understanding, let me think actually. If you're allowed to use it for the quizzes, I would imagine you're allowed to use it for the exam, but I would double check. I would confirm with Shajani. If she also allows you to, then I would ask as well then to use your notes if you're allowed to. But follow through with what I've said thus far. I think like many things I've mentioned will be very applicable, I hope, inshallah, to your lab exam and that you prep and excel well beyond your imagination and beyond expectations. In this lesson occasion. So, inshallah, I hope that helps. We're over half an hour ahead of schedule, technically. Are there any other things? Will you be the one marking? That's also another question. I don't know. I have a feeling it's automated, so I may not actually be marking you guys, unless they tell me essentially like, oh, no, now it's your like we have to essentially start marking. Because to my understanding, your questions are relatively simple, and like as you could see here, they're not necessarily in need for essentially TAs to mark, like short answer questions or like specific detailed questions for with paragraph descriptions. So just think about how you would be able to, bear in mind that if I wasn't able to mark you, or even if I am, how professional, how sophisticated, how detailed would you be, how comprehensive, how critical, how integrative, how applicative, or how like, how diligent would you be for integrating what I've mentioned, right? The I5s, I remember some integration, innovation, or inventions, implication, and integration. These are five things that you guys can excel with, believe me or not. Um, and just as a testament to this, I don't personally like showing people this, but I'll just show it to you guys to like really showcase. Um, Okay, I'm actually a bit sensitive to this, so let's see if I should or not. Okay, I'll show you guys actually. Um, yeah, that's also another point. Yeah, like you want to be diligent for units as well. Sometimes you may forget to convert or include units, or where it's mentioned not to include units, you may have like added a letter accidentally, or maybe used an O instead of a zero. So you want to bear those details in mind. But I say that like I5 approach, how important it is to me, and I hope how it'll be for you guys, is that you know like I am like if you look at my plethora, not plethora, if you just look at my portfolio, it's not like to look at logos and stuff, but I personally believe that all of this is done for the pleasure and sake and from the might of Allah subhanahu wa ta'ala. And I wouldn't believe at any moment that any of this was done by myself. So like me being here at UFT, though, or like being in the past with CSB, Arts and Science Co-op, Sitkins, UHN, Harvard, Johns Hopkins, SHN, Waterloo, ASAB, OGA, the organization that I'm a part of, alhamdulillah, that I founded, that's growing here a lot in size and number internationally. The MSA chapter we have here at UTSC, IB, MIT, COS, which is a program that I'm a part of at the Faculty of Medicine. So if you guys have questions, like let me know at any time. I'm more than happy to support. I'm also noticing my battery is running low, so hopefully I hope you guys enjoy the rest of your prep for the lab exam and are able to excel beyond measure, beyond anyone's vision or imagination, except from the Lord of all dominions. So with this, سبحانك اللهم وبحمدك نشهد أن لا إله إلا أنت نستغفرك ونتوب إليك. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. Jazakumullahu Khairan Katheera. Barakallahu fikum. Have any questions, folks? Let me know. UG is the organization I founded called Our Golden Age with the goal of reaching our golden age once again. And so I hope, inshallah ta'ala, you guys will be a part of it and be the best of scholars, polymaths, and scientists and beyond in your dimensions and disciplines. Barakallahu fikum. Any final questions? I hope my computer doesn't shut off, but Any final questions? Anyone? No problem. And I hope you guys have continually an amazing time at UTSC and beyond. If you have any questions, feel free to reach out. Hopefully, I have my email, mohammed.saeed@utoronto.ca. I also have it incorporated. Sorry. Oh, I think it's going to shut off. So just feel free to email me. You have me on Quirkus as well. Feel free to message me at any time, and I'll be more than happy to help you guys at any time once again. That's Mohammed.Saeed, pretty much my name, at utoronto or mail.utoronto.ca. So, Jazakumullahu Khairan. Barakallahu feekum. Assalamu Alaykum. Take care. You too, Joshua. And likewise, Dorlin and everyone else. Have an amazing time. All the best in this life and the next.